Hello everyone, I'm Junhu. Today, I would like to present to you the paper I have submitted to the iMetaOmics journal, entitled Clostridium difficile and Intestinal Health, Bacteria, Gut Microbiota, and Diet. Clostridium difficile infection is a growing global health threat, presenting significant challenges to public health. Advances in metabolomics and proteomics have enhanced our understanding of human physiology, particularly the influence of gut microbiota and dietary components on diseases like Clostridium difficile infection. This review explores recent insights into Clostridium difficile infection pathogenesis, including the infection cycle, toxin mechanisms, spore structure, germination, and therapeutic strategies targeting spore formation and germination. We also examine the role of gut microbiota and metabolites in Clostridium difficile infection progression, along with the impact of diet on disease pathology through microbiota modulation and immune regulation. We aim to guide the development of precise, individualized dietary strategies to regulate gut microbiota and immune responses, offering innovative therapeutic options for Clostridium difficile infection prevention. This review investigates the potential mechanisms of Clostridium difficile infection, CDI, including the life cycle of Clostridium difficile, and its pathogenicity in the host. This review explores the interactions between the gut microbiome and gut metabolites, and their role in CDI progression. This review explores dietary modulation as a novel approach to restore gut health and mitigate CDI severity. Clostridium difficile is primarily transmitted in clinical settings, particularly among patients undergoing antibiotic therapy for other conditions, leading to intestinal dysbiosis. Simultaneously, spores are ingested via the oral cavity, and under appropriate conditions, they germinate and produce toxins. Subsequently, as the growth phase of Clostridium difficile concludes, the bacterium produces spores and releases them into the intestinal environment. Some spores are excreted in the host's feces, while others persist and proliferate within the intestine. Toxin A and toxin B are recognized by toll-like receptor 4, triggering an intestinal inflammatory response. Frizzled receptor is a well-established receptor for Clostridium difficile toxins, and its activation impedes intestinal damage repair. Additionally, Clostridium difficile binary toxin is a recently identified Clostridium difficile toxin, recognized by toll-like receptor 2, which induces intestinal inflammation. Myeloid differentiation primary response 88, WN family of proteins, RAS homolog family member A, RAS-related C3 botulinum toxin substrate 1, T helper 2. Gut microbes convert primary bile acids into secondary bile acids, which inhibit Clostridium difficile growth and proliferation. Both primary and secondary bile acids can bind to toxin B, inducing structural changes and reducing its toxicity. Cholic acid, chenodeoxycholic acid, lithocholic acid, deoxycholic acid, graptolite signaling peptide, recurrent Clostridioides difficile infection. Furthermore, short-chain fatty acids regulate the host immune response, alleviating Clostridium difficile infection by nourishing the gut, enhancing the intestinal microbiota, and strengthening the intestinal barrier. Acetate prevents Clostridium difficile translocation to extraintestinal organs by modulating free fatty acid receptor 2 expression. Additionally, acetate alleviates CDI by modulating FFAR2, promoting neutrophil recruitment to the site of inflammation, and inducing interleukin-1 beta secretion. Conversely, butyrate alleviates Clostridium difficile infection by enhancing interleukin-1 receptor sensitivity on lymphoid cell type 3 cells, which induces interleukin-22 secretion and host defense peptide production. This review provides an in-depth review of the pathological mechanism of Clostridium difficile infection and the impact of dietary patterns on its progression. Treatments for Clostridium difficile infection, medical interventions to stop the spread and infection process of Clostridium difficile, to develop and apply methods that can neutralize or reduce the activities of toxin A and toxin B in order to reduce their damage to intestinal epithelial cells. Promotes the restoration of beneficial microbial diversity and stability by regulating the gut microbiome to enhance intestinal health.